chemotherapy for leukemia patients currently has a 2/3 remission rate. That means 1/3 of leukemia patients that go through chemotherapy won't go into remission, simply because chemotherapy can't keep up with leukemic stem cell growth. That's over 23,100 deaths that could have been avoided. And for leukemia, this is even more significant, because for acute leukemia, only 24.7% of adults actually survive 5 years after their diagnosis. We aim to fix this problem by reducing the leukemia cell proliferation rate, so that in periods after chemotherapy, the leukemia will actually grow slower, and so chemotherapy will be more effective and it will lead to higher remission rates in the future. We plan to use artificial intelligence to actually find these genetic variants that lead to reduced cell reproduction and map it to the genome in order to validate these findings. So first, we identified the GWAS, or the genome-wide association studies. So these are essentially the studies that we would find specific genetic variants that actually lead to that specific trait. So over here in this example above, you can see that the data points that are higher than the other data points are essentially the genetic variants that will lead to an increase or decrease in that certain trait and will have a much more higher significance than the other ones. And essentially, we use the MR base to actually filter through these different GWAS and find the one that were actually the most significant and most relevant to our findings. And we actually really like the MR base because of how much data it provided with the sample size, the traits, like the name of the trait, and also just the number of genetic variants that it actually tested. And at the ending, we chose 3 different traits to test our thing on, which is leukemia, leukemic stem cell growth, and then cell growth factor receptors, and then apoptotic cells. We used 2-sample Mendelian randomization to actually test this correlative factor between the genetic variant and the 3 factors that I just discussed before. And then we use a random forest algorithm to actually test a causal relationship to actually say that this genetic variant will actually cause this different, this will actually cause increase or decrease in the leukemic stem cell growth. So we technically always try to use values below, p-values below 0.05, just because any values below that are usually considered to be statistically significant, and we filter out the ones that were above 0.05 so that we can show that these genetic variants are the ones that actually have a causal relationship with the traits that are outlined above, and they lead to either an increase or decrease in the leukemic stem cell growth. And lastly, we map it to the genome by using an internet search, and we actually tested out with a bunch of different research papers to see how our gene actually found, tested out with recent findings about leukemia stem cell growth, and just to see whether these findings actually hold true right now. And we found 5 different genes, and all of these things were actually really instrumental in the growth or decline of leukemia stem cell growth. For example, drugs actually decrease SARM1 to decrease the amount of leukemia in certain patients. So this can show that there is actually a causal relationship between these genes and a decrease or increase in leukemia stem cell growth. So for the applications for this, we essentially want to use we want scientists to use our findings and validate them, and use this in testing trials and see whether this leads to higher remission rates in the future. And also, we want to spread this to different tumors diseases like breast cancer and melanoma, since the MR base is such a comprehensive data set, and we can use this for other diseases as well.